Good morning. My name is Mark Pallen. I'm Professor of Microbial Genomics at the University of Warwick in Warwick Medical School. And Jose Gutierrez-Marcos asked me to speak about metagenomics and microbiomes of humans and other animals on this extreme biology course. I'm not able to do that in person because I've got another commitment in Birmingham and it would mean a lot of travelling if I went to Warwick and then Birmingham and then home again. So I'm just going to record this as a short slidecast. Now I have to admit, when I was asked to talk about this on an extreme biology course, I thought it was a bit of a strange thing to do. But actually, as I'll show, I've been able to put together some slides from various other courses, and it's actually not quite such a mad idea after all. Just give you a quick introduction to me. I'm a medic by training. I did my MD and MLC Path at Barts and became a consultant and a lecturer, a senior lecturer. In my mid-30s I did a Wellcome Trust Research Leave Fellowship and did a PhD full-time. I was on University Challenge while I did that. And since then I've really charted the course away from my medical roots, and I've now become a full-time academic, and I don't actually have any clinical practice anymore. I've had chairs in Queen's, Belfast, Birmingham, and now most recently in Warwick since April 2013. And my research interests straddle bioinformatics, bacterial pathogenomics, genomic epidemiology, and metagenomics and microbiomes, of which we'll say more about in a moment. You can find me on Twitter, you can find me on YouTube as well, and if you're interested in any of the things I say here, there are many talks that cover many of these issues in much more detail available on YouTube, as I've made all of my teaching and most of my research seminars available on YouTube over the last couple of years. And if any of you are interested in coming to do projects with me, PhD projects or final year student projects, or want to contact me or ask me questions, you can contact me on m.pallen@warwick.ac.uk. Now, let's just focus in first of all on the microbial universe as it applies to us. Now, our bodies are like mobile, warm-blooded coral reefs. They're very rich in microbial biodiversity. Okay, we have to look down the microscope to see it, or we have to do sequencing to see it, but it is a truly rich universe down there in the microscopic realm. And it's a kind of truism now, it's often repeated, that there are more bacterial cells associated with the human body than there are human ones. Usually it's said by a factor of 10, although I think the evidence for that is not exactly clear. But certainly we have this very rich set of organisms. This is known as the human microbiota. Other organisms, other animals have a similar kind of microbiota. On this view, we humans can be viewed as superorganisms, built not just from our human cells, the ones you learn about in your cell biology classes, but from all those microbial cells as well. Julian Davies made this kind of slightly quirky comment here, said that humans are large, highly complex microbial communities attached to some relatively uninteresting organic matter. And there's been a great deal of interest in this. There's this project called the Human Microbiome Project in the US, launched with great fanfare a few years ago, which is looking to catalogue all the organisms associated with the human microbiome and look how this microbiota changes in health and disease. It's said that this is in effect a multicellular organism. It's as complex as the liver. Probably in terms of weight, it's about the same size as the liver in terms of if you weighed all the bacteria associated with you. And it's been described as the third human genome, after the human nuclear genome and the human mitochondrial genome. And sometimes the term microbiome is used to describe this community, particularly this community and all of the genomes and genes that go with it. And the human microbiome has at least 100 times more genes than the human nuclear genome. So in that sense, it's not really hyping it up to say that this is a largely unexplored third genome. Now, just to give you some definitions of terms, we use the term commensal for any of these organisms that live in that microbiota, normally found on external body surfaces. We take a topological view when we're talking about the human body. So as you can see in this little figure here, this is what a human looks like topologically. The gastrointestinal tract, all the things inside the lumen of the bowel are actually outside the body from a topological viewpoint. We can view it as this kind of load of flesh surrounding a tube, but nonetheless that tube is out exposed to the outside world. That total commensal population constitutes the human microbiota. You sometimes see in old books and old articles the term normal flora when people thought that bacteria were closely allied to plants. We now deprecate that term and we prefer to use the term microbiota. This microbiota includes bacteria, protozoa, fungi, archaea, metazoa, viruses, all manner of microscopic organisms. And its composition varies from one individual to another. There are some species that are carried away only transiently. If you eat some lettuce, it's probably got a few soil bacteria on it, but those will be destroyed very quickly and lost. And then there are many organisms that are fairly permanent, and you'll be carrying them for weeks, months, sometimes even years. And it's actually very difficult to alter the composition of the normal microbiota of the gut of a healthy individual. If I fed you an E. coli strain, it would be very hard for me to displace the E. coli that already lives within your bowel and replace it with that strain without doing some manipulation of the microbial ecology first. Now, I'm not going to go through all of these points one by one, but there are lots of beneficial effects of the human microbiota. It's really part of us and it's necessary for normal development. It provides a kind of blanket against colonization by other bacteria that might be harmful, so-called colonization resistance. It's involved in assimilation of nutrients and breaking down some of the components of our diet. But of course, the human microbiota can also be involved in disease. You can get commensals causing localized disease. So something like Streptococcus mutans, a normal part of the microbiota in the mouth. But if you eat sugar, it then makes acid, and that causes dental caries. Streptococcus pneumoniae, another organism that lives in the upper airways, nasopharynx, without doing any harm. But if it spreads sideways into the middle ear or into sinuses, it can cause inflammation and disease. Obviously, this microbiota can escape to abnormal sites. So if you burst your appendix, all of those organisms that normally live inside your bowel, topologically outside of the body, are suddenly inside the body, and they cause peritonitis, which can be life-threatening. And similarly, things can track up the biliary tract and cause cholecystitis, cholangitis. And urinary tract infections usually are caused by organisms coming from the gut. The normal microbiota in the gut, doing no harm there, when they get into the urinary tract, then can establish themselves as pathogens and cause disease. In fact, over the recent years, recent decades even, we've now recognised that many of the organisms that form part of the microbiota can cause disease in certain situations. So, for example, Staphylococcus epidermidis normally lives harmlessly on the skin, can be very problematic if it's in an immunocompromised host, or more particularly in this case with Staphylococcus epidermidis, if there's foreign material like catheters or artificial joints or artificial heart valves or pacemakers, any bits of metal or plastic in the body, if these organisms get onto them, they can form what we call biofilms and become very difficult to eradicate. It's clear also that in recent years it's become clear that there are many interactions between the host and the microbiota that actually tip the balance between disease and health in conditions that are not normally thought of as infections. So there's this idea of the hygiene hypothesis that all those bacteria that we're exposed to, that we're eating and drinking, tune our immune system, and the lack of exposure to those can be perhaps seen as a cause of things like allergies like asthma and eczema. It's also clear that the gut microbiota, which is the largest microbiota associated with this, has been implicated in pathogenesis of bowel cancer, inflammatory bowel disease, irritable bowel syndrome, diabetes, and perhaps most notably in the control of weight. And so there's now evidence that the gut microbiota can play an influence in whether you are obese or whether you're lean. There are differences in composition in obese individuals and lean individuals and when people diet. And even if it's even been shown that if you transfer the microbiota from an obese mouse to a lean mouse, it leads to that lean mouse gaining excess weight. And I think also vice versa, if you put lean microbiota into obese mice, they lose weight. So there's a great deal of interest in this area. Here's one particular article, it's a few years ago now, but there have been quite a few since then. This is one that really led launched this kind of particular interest in the gut microbiome, showing that you can actually define an obesity-associated gut microbiome. Now, moving away for a moment from the gut microbiome and other microbiomes, to just look at virulence. So when organisms stop being commensals and actually become pathogens, what's going on? And why is this important to extreme biology? Well, we now view virulence as a multifactorial process. We don't just say, oh, an organism produces a toxin, and that's that. We kind of recognise that a bacterial army invading you or colonising you, like a human army, needs more than just its firearms to enter and secure enemy territory. And we recognise that there's a whole program of events going on. So if you swallow some E. coli, it's going to have a very different environment in your mouth than what it meets in the stomach, and then what it meets in the small bowel, and then what it meets in the large bowel. And so that E. coli will have to deploy different defences, different repertoires of genes and surface proteins and other components at different times in its passage through the gastrointestinal tract. In fact, if we look at the way in which we are protected against infection, many of the defences are physiological. So the acquired immune response you hear a lot about in immunology, but there are a lot of things going on before you even get to that stage. And many of these responses are actually there to stress bacteria and to give them a hard time. Try and wash them out of the body, try and decrease their growth. Now, this is a slide I pulled out of a lecture I gave on the pathogenesis of infection more generally, and rather fancifully I broke down all the various steps in infection to terms that could start with the letter S. And what I'm going to focus on here is survived stress, because that's one particular feature that we recognise in bacterial infections and is obviously relevant to extreme environments and extreme biology. So in addition to nutrient-limited stress, so they're starved in certain compartments, bacteria face many other stresses. The key one here is obviously acid stress within the stomach. The stomach really is an acid bath, and all that acid is there to destroy those bacteria that are coming in. And so very few organisms can actually survive passage through the stomach. But if you think about it, everything that's in the lower bowel and the large intestine or small intestine has to have got through that acid bath to get there. It's recognised that fever is a kind of form of heat shock, and actually it is a way of actually tuning down replication of the microbial invader while also stressing the host, but it's a battle between the two. And release of oxidative stress, reactive oxygen molecules, reactive nitrogen molecules as well within phagocytes. These are very stressful events for the bacterium that's actually been encountered. And most bacteria actually don't survive, they get destroyed by these things. So to be a successful pathogen, you have to have a mechanism for overcoming those things. Interestingly, when people started to look at what were the dominant antigens, when you look at what antigens do we make antibodies against, for example, they found stress response proteins, particularly chaperonins, feature as those immunodominant antigens, and it's a kind of measure of the fact that these organisms are under stress as they're causing disease. Some proteins are involved in detoxification of stresses like superoxide dismutases, for example, can play a role in virulence. And to come back to the acid stress of the stomach, we know that this is important in disease because if we remove acid, so you can do this by giving people acid suppressant medication, the infectious dose you need for an enteric pathogen like Vibrio cholerae or E. coli to establish disease is much lower than it would be in a normal individual. In fact, let me just say a few words about a very interesting organism here to come back to Pylori, which is actually adapted as an extremophile, if you like, an acidophile to live within the stomach. This is actually associated with gastritis, inflammation of the stomach, duodenal ulcers, and gastric cancers. And it can colonise the antral epithelium of the gut, despite the fact that it's a kind of acid bath. It's actually an organism that's disappearing as we're getting better hygiene and more use of antibiotics. The story behind this, the history of it, is quite interesting. It had been observed many times before, but never followed up. And the conventional thinking was, people just said, oh, the acid environment is too extreme, nothing can live there. But it was first, the first time it actually came to attention and something was done about it. That train of events started in 1979 when the Australian pathologist Rob Warren here on the left saw the organism down the microscope, and then he started working with his physician Barry Marshall, and they tried to grow the organism. And there was a bit of serendipity here, because as they were trying, they were just leaving plates up overnight. But one time they left their plates up over 5 days, over an Easter weekend in 1982, and that was long enough for the organism to actually start growing. And they published a letter in the Lancet about this, and it was met with great skepticism to begin with. People said, oh, well, these organisms aren't there, or they said, well, right, they're there, but they're not doing anything, they're not causally associated with any disease. And interestingly, we now recognise that they are, and they have all these virulence factors. One of the most dominant virulence factors is urease, which breaks down urea into ammonia, and that neutralises the acid, and it also gives the bacteria a way of scavenging nitrogen. And I played a small role in this story of urease, in that I was one of the members of the team that actually first cloned and sequenced the urease genes from H. pylori a long, long time ago. There are various other toxins there, but none of those so closely related to stress and extreme environments. Coming back to the history, because nobody believed Marshall, he actually in 1985 drank a beaker full of H. pylori culture and developed nausea and vomiting, and gastroscopy confirmed he got gastritis. So a rather extreme thing to do. Shortly after that, antibiotics were shown to be effective in the treatment of gastritis and prevention of relapse of duodenal ulcers, when those antibiotics were tailored to H. pylori. And in my lifetime, this has been one of those kind of miracles, if you like. Warren and Marshall rewrote the textbooks with reference to what causes gastritis and peptic ulcers, and we now have these simple, cheap antibiotic regimes replacing what was torturous surgery when I was a young man, when I first came into medicine. The kinds of operations surgeons had to do for intractable duodenal ulcers were extreme and horrible. And quite justifiably, they got, in 2005, awarded the Nobel Prize in Medicine for their efforts. Now, just to turn back to microbiomes, one of our interests is in what we call shotgun metagenomics, where those microbiomes are very complex. They've got lots of different organisms in them. You can extract DNA, though, from a sample from a human, say a fecal sample, and then just shotgun sequence all of those organisms together. All the sequences from those organisms en masse is called the metagenome. It may contain dozens, hundreds, maybe even over 1,000 genomes in the most complex metagenomes. And then the trick then is to assemble and analyze that data. And that can be quite tricky, because it is very complex data. In my group, we have an interest in the chicken cecal metagenome and microbiome. We've taken total DNA from a chicken cecum and sequenced that using an Illumina HiSeq, a high-throughput sequencing instrument, and we analysed those sequences to try and work out what was going on in that microbiome. Martin Sargent, the postdoc in the project, was able to reconstruct some of the genomes of the most abundant bacteria without having cultured them, just sequencing the material. And he found many genomes in his top 10, top 20 list that actually showed limited identity to known species. So here you can see an alignment between the C. jejuni that came out, Campylobacter jejuni that came out of the metagenome with a genome sequence strain. And here is his top 10 or top 20 list of bacteria that he found. And he's listed the closest match here, and sometimes the closest match, there's similarity at the level of 100%. But in other cases, you can see the closest match has a similarity of less than 90%, and therefore it is effectively an unknown, previously undescribed species. And remarkable that these are in such a commonplace place as the chicken gut. Martin Sargent's been looking for all sorts of enzymes in this microbiome, enzymes that might be useful in terms of chicken feed supplements to allow chickens to recover more nutrients from their diet, or in other situations as well. And he's even been able to reconstruct some of the operons through his assemblies that are involved in handling complex carbohydrates in these organisms. And this is giving us new insights into the way in which carbohydrates are handled in the gut microbiome of the chicken. Just going to finish up now. So we mentioned shotgun metagenomics, but one particular interest of mine is the idea of diagnostic metagenomics. So we can use shotgun metagenomics in terms of microbial ecology to find out what's there and so forth. But my interest is a more simple one. So in conventional microbiology, we take, say, a fecal sample, we plate it out and we grow colonies, and then we might pick those colonies and sequence genomes from them. But why can't we just go directly from that fecal sample to detecting a pathogen by just extracting DNA and sequencing it? And that's the question that I posed in my group and we've been trying to answer. Here's another slide that shows a similar kind of thing. So conventional approach is microscopy and culture. We get various organisms growing in pure culture, subculture them, we then sequence them or we then identify them. It's a very long drawn out process, but it does have the advantage that you don't know what you're going to grow. You get all sorts of organisms growing there. In the last 20 years or so, there's been advances in molecular diagnostics where people do things like PCR to detect DNA from a particular pathogen and thereby detect the pathogen in the sample. And this has many advantages. It's simple workflow, sensitive, you don't have to culture. But the problem is you have to define the target in advance. So you're only finding what you're looking for. And if you're looking for multiple things, it's a lot of work to optimize the assays multiple times over. And so what we're interested in doing is seeing if we can do diagnostic metagenomics, where we can just do shotgun sequencing. So we'll analyze the sequences. We can throw away all the unwanted sequences from things that aren't pathogens, and then focus down on the pathogens and see if we can actually reconstruct information about the pathogen and give us access to genome level data about epidemiology, virulence, antibiotic resistance that we wouldn't get if we were just growing organisms. Now, time is short, so I'm not going to go into any details here, but just suffice to point in the direction of two recent publications that we've had in this area. One where we looked at stool samples from an outbreak in Germany, and we were able to show that we could get the outbreak strain genome directly from the metagenome of fecal samples without doing any culture. So that was an interesting finding. And then more recently, we've applied this approach to 200-year-old mummified lung tissue, and we've been able, through metagenomics, to actually detect tuberculosis and mycobacterium tuberculosis in this historical material. And this opens up the potential of actually unpicking the epidemiology of tuberculosis, the spread of tuberculosis, 200 years ago in Hungary, when TB was much more prevalent in Europe than it is now. That's me finished. And I'll call it a day there. Thank you very much. Email me if you have any questions. Thank you.